Introduction. When we perform the elemental analysis of living and non-living, we get the similar results. The only difference is in their relative abundances. To perform the elemental analysis, we need to crush the subject with mortar and pestle using trichloroacetic acid. Next is the amino acids. Amino acids are the compound containing amino group and carboxyl group on the same carbon, that is alpha carbon. They are also called substituted methanes. There are many amino acids, but only 20 of them are found in proteins. Amino acids can be acidic like glutamic acid, basic like lysine, and neutral like valine. They can also be aromatic like tyrosine. The next topic is lipids. They are generally water insoluble. They can be fatty acid, glycerol, or phospholipids. Fatty acids are the carboxyl group attached to the R group. They may be saturated or unsaturated, for example, palmitic acid and arachidonic acid. Now, glycerol. They are the trihydroxypropane. When glycerol is esterified with fatty acids, they give fats or oils. Oils generally have lower melting point. Phospholipids are the lipids which have phosphorus in them. They are found in cell membranes. For example, lecithin or neural tissue has more complex phospholipids. Next is nitrogen bases. They are of two types, purines and pyrimidines. Purines include adenine and guanine. Pyrimidines include thymine, cytosine, and uracil. Next is nucleosides. When sugar is found attached with nitrogen bases, they form nucleosides. For example, adenosine, guanosine, thymidine, cytidine, uridine. Next is nucleotides. When phosphate group is attached with nucleoside, they form nucleotides. For example, adenylic acid, guanylic acid, thymidyllic acid, cytidyllic acid, and uridylic acid. The next topic is metabolites. There are two types, primary metabolite and secondary metabolite. Primary metabolites are those which are found in animal tissue, and secondary metabolites are not found in animal tissue. Primary metabolites have known roles to function, while we do not know the function of secondary metabolites. Here is a table of secondary metabolites which includes pigments like carotenoids and anthocyanins, alkaloids like morphine and codeine, terpenoids like monoterpene and diterpene, essentials oil like lemongrass oil, toxin, abrine and resin, lactins like concanavalin A, drugs like vinblastine and curcumin, polymeric substances like rubber, gums, and cellulose. Next is biomolecules. Biomolecules are of two types, micromolecules and macromolecules. Micromolecules, they weight less than 800 Dalton. They are the contents of acid soluble pool. The acid soluble pool represents roughly the cytoplasmic composition. Example of micromolecule is amino acids. Macromolecules, they weigh more than 1000 Dalton. They are the contents of acid insoluble pool. They include protein, nucleic acids, polysaccharides, and lipids. Thank you.